Good morning, everyone. If you're loitering out in the hallway trying to decide which workshop you should attend right now, we think workshop 34, ions, fluid and mucus transport, will be a fantastic selection. So if we could all take our seats, silence our phones, get back up here so I can be heard. I don't have to kneel down today. That's a nice change of pace for me, for those who have been in other sessions. On behalf of my co-chair, Dr. Vijay Kumar, we'd like to welcome you to this workshop. Today, our goal is to foster a discussion on real-world clinical responses in the forms of inflammation, infections, mucus clearance and viscoelastic properties to ETI, and then look at some new therapies that are in the pipeline coming forward that could also be beneficial to CF patients. And with that, I'm going to hand it off to my co-chair.
Thanks, David. Hi, everyone. When we did this exercise of choosing abstracts, we said we probably want a representation of what's happening in the real world with ETI, what it fixed, what it didn't fix. So we picked a combination of human studies, in vitro studies, and animal studies. So looking forward to this. Our learning objectives for this session are to describe the longitudinal effects of ETI on airway inflammatory markers, infection progression and changes to sputum rheology, identify in vitro factors that contribute to the persistence of inflammation and infection despite ATMT from the sinonasal airway epithelial cells and the lung airway macrophages, and to highlight the effects of CFTR mRNA transfection on mucus and mucociliary clearance and the ferret airways as we look forward to moving from ETI to genetic therapies. So with that, I would like to invite Sam Nolan, who's our first speaker. He will be talking to us about the RECOVER study. Which focuses on airway infection and inflammation in people with CF treated with ETI.
Cool. Okay. Thank you very much for the introduction. So I'm Sam Nolan. I'm a PhD candidate from the Queen's University of Belfast. It's an honor to be here today. This is actually my first conference, so it's very exciting. I'm here today to talk to you about the RECOVER study, and many of you may have heard the acronym RECOVER thrown around over the past couple of days, and I'm going to talk to you specifically about airway infection inflammation in a cohort of these patients treated with ETI therapy. So these are my disclosures. There are none. And first of all, just a overview of the RECOVER study, for those of you who do not know. So RECOVER stands for Real World Clinical Outcomes with Novel Modulator Therapy Combinations in People with Cystic Fibrosis. And it is a multi-center cohort study which takes place in 7 pediatric and adult sites across Ireland and the UK over a 3-year period, in conjunction with the CF Registry of Ireland and the UK. And just a bit of rationale and overview of the study. So RECOVER is set up to be a real-world non-interventional study examining the impact of new triple combination CFTR modulator compounds and the compound ETI on clinical outcomes in people with cystic fibrosis through a large biobanking trial. The introduction of ETI for a large proportion of people with cystic fibrosis represents a new era in the management of cystic fibrosis. This real-world non-interventional study, like RECOVER, is an important complementary source of knowledge on the impact of treatments with people with cystic fibrosis. To do this, we're collecting an extremely comprehensive data set and a bank of human samples with longitudinal follow-up over 3 years to maximize research benefit for these patients. So this is our study design. To date, 145 homozygous patients for F508del and 59 heterozygous with a minimal function mutation have been recruited onto the study. Today, I'm just going to be talking about the patients aged over 12. All of these patients are randomized into the standard testing group, where we collect a number of samples, and a further group of those are then randomized into the advanced testing group, where we collect ultra-low dose spirometry-controlled CT scanning, sputum collection, and nasal lavage. This table, I don't expect you to see or read all of it, is just a number of the samples and markers we're collecting in this study. As you can see, there's a lot going on, so we're able to collect a lot of real-world data about these patients. Again, I'm just talking about airway infection inflammation, so I'll just be discussing the airway sampling, nasal lavage fluid, and sputum. The aims of this study as a whole determined the effect of ETI therapy on pulmonary function, spirometry-controlled CT scores, as well as airway infection inflammation, which is where I come in. Then also nutrition, gastrointestinal systems, gut inflammation and pancreatic functions, and adherence and understanding of overall CF treatment. So moving on to some results then. So first of all, this study was kind of designed around airway samples being sent away to local laboratories. And here we have the samples that we took for measuring infection in these patients over the 24-month period. We collected throat swabs and sputum where available, but as you can see, which is sort of the trend, and I'll be discussing this a bit more, as time goes on, we are able to collect less sputum, so we use throat swabs. This histogram shows a proportional prevalence of the 7 most abundant bacterial and fungal isolates from the CF airway samples that we collected, as well as how many patient samples were available and how many organisms were identified at each time point in total. What we're able to see is by using chi-squared analysis, at the 12-month period, we see a significant decrease in the amount of Staphylococcus aureus and Pseudomonas aeruginosa detected in throat swabs and sputum samples by these patients. There's the actual chi-squared data if you'd like to see. These continue to trend down at 24 months. However, we can't really be sure on significance as the number of samples that we have at the moment at 24 months doesn't match up to 12 and 6. For Streptococcus, Haemophilus in Canada, all of these isolates trend downwards, however, in a non-significant fashion. What's interesting is for the other and commensals, these tend to stay the same over the 24-month period. We then classified our infections by the Leeds criteria. So for anyone that isn't familiar, chronic infection is classified by a number of samples of over 50% of samples in the previous year of testing positive for a certain isolate, intermittent by less than 50%. A patient is identified as free of infection if they've identified with a pathogen before, however, not over the past 12 months, and never is obviously never. And what we see is a proportion of patients in each classification and over the 24 months, and we see at 12 months, we get a significant increase in the number of patients in the free category. And at 24 months, we see a complete removal of all patients classified as having a chronic infection. So it goes from 21 out of 124 at the baseline to 0 out of 27 at 24 months. So we thought this was really interesting. And it's important to note that antibiotic usage by these patients also decreases over time. So this is some data that was presented at ECFS this year. And what we can see is for all of our patients, whether antibiotic usage was intravenous or oral, the amount of antibiotics significantly decreases over time. So this means that the infection class is lessening and number of isolates lessening was despite antibiotic usage also decreasing. So some key points from this bit that I've just presented. So Pseudomonas aeruginosa and Staph aureus infections significantly decrease after 12 months of ETIA treatment, and infections continue to trend downwards after 24 months. Other microbiological isolates and commensal microorganisms are at similar levels after 24 months, and the number of patients free from chronic infections significantly increases at 12 months, while those chronically infected became 0 after 24. And this is despite reduced antibiotic usage, which is really cool. So moving on to some inflammation data. We collected sputum as is the standard, but we knew that sputum would be kind of hard to collect after commencement of ETI therapy. So we also collected nasal lavage fluid by flushing the nasal cavity with 10 milliliters of saline water and collecting that. The issue here is that is NLF the answer to our sputum woes, if you like, and you may see or may not see if it is the answer in the next sort of couple of slides. So the first marker we wanted to look at for inflammation was interleukin-8, and interleukin-8 is one of the major mediators of inflammatory responses. It's secreted by several cell types and functions as a chemoattractant, mainly for neutrophils. And by measuring isolate concentration by ELISA method, the ELISA kits were provided by Biotechne, we see a significant decrease in the amount of isolate detected in sputum samples at 6 months. It looks like it's possibly increasing at 12 and 24 months, but the number of samples there makes it very hard to say anything at all. We see a non-significant decrease in the amount of interleukin-8 detected in nasal lavage fluid samples at 12 months. However, yeah, it's not significant. We can't really be sure. So we're not really sure if nasal lavage fluid there is the answer for this. We then looked at neutrophil elastase. This paper by Myra Hamblett, which I'm sure many people are familiar with, showed that neutrophil elastase concentration is directly related to lung function in the CF patient. So we looked at activity using fluorometric methods. And what we see is that at 6 and 12 months in the sputum and nasolavage fluid, respectively, we see a significant decrease in the amount of any activity, free any activity recorded. As a number of these measurements that measured in the change in RFU are 0, it then moved on to some more chi-squared analysis, and we see the exact same pattern. By measuring present versus absent any activity, we see, again, significant decrease at 6 months and 12 months in the sputum and nasolavage fluid, respectively. In the 24-month nasolavage fluid samples that I've measured so far, it continues to be 0. I then looked at cathepsin S. So cathepsin S is sort of my PhD baby. A lot of people haven't heard of it. And it's lysosomal cysteine protease that's been implicated in the pathogenesis of many lung diseases like COPD, ARDS, and cystic fibrosis. It is involved in many different things, and it's predicted to be involved in some others. So it's involved in MHC2 mediated antigen presentation, modulation of immune response, and inflammation and tissue remodeling. Previous work in my lab has shown in a murine model, if you knock out or inhibit cathepsin S, it leads to reduced neutrophilic inflammation of the lungs and reduced neutrophilic burden. And by measuring cathepsin S concentration in these samples, we get an interesting kind of story where it doesn't really change in the nasolavage fluid by ELISA, and it has a non-significant decrease at 6 months. What this means, not really sure. But the next step is to look at activity in these samples. We've also then looked at some initial proteomic data. So far, 8 nasal lavage fluid samples have been sent off for analysis. And what you can see here is these samples that are, the 2 dots that are close together are technical replicates, and each color is a patient. And what we're seeing is for some of our patients, like in green and in orange there, are really big differences on the pre and post ETI treatment level, as these samples are at baseline at 12 months of ETI treatment. So more analysis will be done in the future. However, this data has only become available from the last week, and I still need to go through it all. But then if we look into, so this is our PCOA plot, if we look into our volcano plots, we can see a number of proteins are significantly different. So for a number of patients, these are 2 different patients, we see over 300 significantly different proteins that were found, with 1,300 identified. Some interesting proteins that came up as significantly different. So the serine leukocyte protease inhibitor SLIPI was upregulated in these patients, as well as neutrophil defensin 3. Defensins are a family of microbicidal and cytotoxic peptides thought to be involved in host defense. They're abundant in the granules of neutrophils. And then cystatin A and cystatin M. Oh, can I go back? Yes, there we go. Cystatin A and cystatin M, they're antiproteases that are downregulated in lung diseases. So it's kind of cool to see those go back up again in these patients. Downregulated, we have MUC5AC. MUC5AC is a component of mucus, as many of you will know. Interesting one I pulled out was Lumicin. So this promotes joint fibrosis through TGFB signaling, and it's elevated in the lungs in ARDS patients. So I thought this was interesting. It was going down as it promotes fibrotic activity. And also angiotensin. So angiotensin is a potent inhibitor of NO synthase, and it acts in regulation of blood pressure. It's reduced signaling of angiotensin is actually beneficial to CF lung function. So it's cool to see that going down as well. But further work is ongoing, and all of our samples will be eventually sent for proteomic analysis. So in summary, treatment with ETI therapy has led to significant decreases in infection and papular cystic fibrosis age 12 and older. It's led to significant decreases in markers of inflammation, ILA and NE, at least in the sputum. And sputum samples will be harder to ascertain, as you can see in these slides, and we need to look for alternatives. So whether nasal lavage fluid is the best thing for this, we're not sure, as inflammation and infection can be compartmentalized, and nasal lavage fluid could be affected by sinusitis and nasal polyp status. And some future directions for the study. So I'm doing the lab work, so I still need to do some of the 12-month sputum samples and 24-month nasal fluid samples as well. We're then continuing to expand our proteomic analysis, and metagenomics on samples from each time point will also be ran. So we're going to be able to do a deep dive analysis between our proteomics and metagenomics. And as well as the recover extension study. So we're collecting annual samples on some participants for the next 5 years. So that should provide some really interesting real world data as well. And that's the end of my talk. I'd just like to thank all our collaborators and my PhD supervisor, Cliff Taggart there. And a number of, so this is the AAR lab in the Wellcome Wilson Center for Experimental Medicine and the Recover Study team. And a number of the institutions we work with. So thank you very much for listening. It's been an honor. And if you have any questions, my email's there.
Trying to figure out the microphone. Okay. I think I have a PhD. Okay, so in your PCA plot, you were saying that some samples, like pre and post CTI, kind of were far from each other, and other ones were kind of close to each other, pre and post. Do you have any kind of correlation with like clinical parameters that can explain, the ones that are together and the ones that are like separate? Pre and post.
So this data is really initial. So we only got this data last week, and this is just the first 8 samples we've run. It is in the back of my mind to start looking at clinical parameters. So the genotype, sex, age, as well as their infection status. I think it'd be really interesting to compare their infection status of these patients to what we're seeing on the proteomic data. But yeah, this is very initial so far.
Also, as the one doing the plating here, so did you look at MRSA and mucoidy in those Pseudomonas strains? Are we seeing differences there?
I'm not sure. So I didn't specifically do the microbiology. I just have the data, but I can double check that. Thank you for the question.
Hi. I know you didn't want to focus on it, but I saw in your table there was some liver parameters. Would you like
Sorry?
liver?
Liver? Oh, yes, sorry. I don't have the liver data. I'm sorry.
Okay.
I'm just the infection and inflammation guy.
Does this? Oh, cool. No light, but it works. So you have 5AC. How about 5B? Did you guys get data from? Did you get data on MUC5B as well?
It's come up in a couple of the patients, just not the 2 that I showed there. So MUC5AC and MUC5B are both going down in some of the 8 patients that we've looked at there.
How about the ratio between the 2? Is it?
Oh, you're testing my knowledge there. Again, this is very. I was doing this analysis like the day before I flew out here. So yeah, it's very initial, but I will look into it.
All right. Well, if there are no more questions, we'll thank our speaker and invite our next up. Thank you. All right, so it's our pleasure to welcome Aditi Kupa from the Berlin group. If I mispronounce that, I'm horribly sorry. But she's going to talk about something that I love a lot, mucus viscoelastic properties, as part of their ETI studies. So take it away.
Okay. Or did you want just adjust a bit? Yes.
Yes, thank you for the introduction. I'm very happy to be able to present this collaborative project here at NACFC. I have no disclosures. So the data I will present today are collected as part of a national German multi-center study, Modulate CF, a very comprehensive, prospective, observational post-approval study. Adult and pediatric CF patients are examined before initiation and under modulator therapy at regular intervals. Outcome measures comprise biomarkers of CF function, such as intestinal current measurements, nasal PD, and sweat chloride, novel clinical endpoints such as multiple breath washout and MRI, sputum properties, including bacterial colonization, inflammation, and mucus viscoelasticity, as well as questionnaires assessing quality of life and mental health of patients. Today, I will focus on the mucus properties of adolescent and adult patients during the first year of ETI treatment. So, since I will talk about the changes of viscoelastic properties of sputum due to ETI treatment, I'm going to give a quick introduction into the importance of sputum viscoelasticity. In a healthy lung, airway mucus consists of around 97% water and around 3% mucins and other molecules. And it has an important role as it keeps airway surfaces hydrated and entraps inhaled pathogens and particles that are constantly cleared from the lung by mucociliary clearance. The CFTR chloride channel is essential for the formation of this thin protective mucus layer, as it has an important role in fluid secretion. In the healthy lung, the secreted mucin monomers form a gel-like multimeric network with specific viscoelastic properties. And the amount of mucin cross-linking and concentration can influence these viscoelastic properties, and they're important for effective mucociliary clearance. So in CF, we know that there is a hydration problem since the deficient CFTR-mediated secretion of fluid onto airway surfaces leads to formation of hyperconcentrated mucus. And more recently, we now know that there's also a cross-linking problem. So the hyperconcentrated mucus with increased viscoelasticity impedes mucociliary clearance and then leads to a vicious cycle of chronic bacterial infection and chronic airway inflammation, and then resulting increased oxidative stress leads to further increase of mucus viscoelasticity via increased disulfide cross-linking. So to sum up, dehydration and increased cross-linking lead to abnormal viscoelasticity and impaired mucociliary clearance, resulting in mucus plugging, which you can see in this image. Previously, using nasal potential difference and intestinal current measurements, we showed increase in CFTR function in different organs when comparing baseline measurements to 3 months of ETI therapy. You can see nasal potential difference measurements on the left showing an increased CFTR activity to around 45% of normal CFTR activity. And in the figure on the right, you can see intestinal current measurements to around 40% of normal CFTR activity in intestinal current. So this level of improvement of CFTR activity is correlated with reduced mucus plugging after ETI treatment in lung MRIs. In the representative lung MRI images on the left of a CF patient at baseline and after ETI treatment, you can appreciate both reduced wall thickening and bronchiectasis, as well as reduced mucus plugging. So this data shows the drastic reduction of mucus plugging. As I've shown, studies had demonstrated that ETI therapy improves CFTR function and reduces mucus plugging, but downstream effects of restoration of CFTR function by ETI, such as viscoelastic properties of airway, mucus, and chronic airway infection and inflammation, had not been studied yet. So the aim of our project was to determine longitudinal effects of ETI on airway mucus rheology, microbiome, inflammation, and proteome through the first 12 months of therapy. You can see the clinical characteristics on the right in the table, where we looked at 10 healthy subjects and 75 CF patients with at least one F508del allele at baseline and then after 1, 3, or 12 months. So in this talk, I will focus on these viscoelastic properties measured via rheology and only briefly go over other changes, but feel free to see poster 66 by my colleague Laura Schaub for more information on these data. So oscillatory rheological measurements were performed with a rheometer using a 20 millimeter and 1 degree cone and plate geometry and a protocol that was described previously in our lab, and able to show differences between healthy and CF in elastic modulus G prime and viscous modulus G double prime. The measurements were done at 25 degrees Celsius. Amplitude sweep was performed at 1 Hertz and from 0.01 to 100% strain. And frequency sweep was performed at 2% strain and from 10 to 0.1 Hertz. So these measurements were then used to calculate elastic modulus G prime, viscous modulus G double prime, and mesh pore size, which are all common parameters to analyze viscoelasticity of sputum. With the protocol I just explained, we performed these rheological measurements to determine G prime and G double prime of sputum from healthy controls and CF patients at baseline and 1, 3, and 12 months post ETI therapy or during ETI therapy. In both groups, G prime dominated over G double prime, indicating gel-like behavior of airway sputum. And both G prime and double prime are increased at baseline compared to healthy. There was no change after 1 month of ETI therapy. And after 3 months, then both G prime and G double prime were significantly reduced and stayed reduced after 12 months. But both are still increased compared to the healthy controls. We also analyzed change in mesh pore size. And to visualize mesh pore size of sputum, you can see 2 scanning electron microscopy images on the left of healthy and CF sputum. The sputum mucin network forms these pores, which you can appreciate in these images. And higher concentration and higher cross-linking lead to smaller mesh pore size, which is another way to show change in mucous network properties. Mesh pore size was then calculated with our rheological measurements, and you can see this data here, where mesh pore size of CF sputum is decreased compared to healthy and increased after 12 months of ETI treatment compared to baseline. Now I will show some additional data, and as I've mentioned, you can see poster 66 for more detail. So ETI treatment shifted airway microbiome composition toward healthy. ETI, in this figure here, you can see relative abundance of the most abundant bacteria. And you can appreciate Pseudomonas relative abundance in red down there is reduced by ETI treatment. So compared to baseline, relative abundance was unchanged at 1 month and then decreased at 3 months and stayed low at 12 months. Furthermore, ETI treatment is increased alpha diversity. So alpha diversity was decreased at baseline compared to healthy and then increased during ETI treatment. To analyze airway inflammation, we measured IL-1 beta and IL-8 concentration, which are both increased again in baseline CF sputum. IL-1 beta was reduced at 1, 3, and 12 months post-ETI, and IL-8 was reduced after 3 months, both indicating decreased inflammation in airway after ETI therapy. To analyze protease burden, we measured free neutrophil elastase activity in sputum and membrane-bound neutrophil elastase on sputum neutrophils, which are both elevated in CF baseline compared to healthy. And ETI significantly reduced the activity of free NE in sputum after 1, 3, and 12 months, and membrane-bound NE activity was significantly reduced at 12 months. So to come to the conclusion, I've shown that ETI therapy improves viscoelastic properties from CF patients toward healthy levels. elastic modulus G prime and viscous modulus G double prime were significantly reduced after 3 months. The improvement of viscoelastic properties is associated with improvement in inflammation. And to conclude, these positive effects were sustained through 12 months, but did not reach healthy levels. So therefore, the residual disease activity we can see under these highly effective CFTR modulator therapies indicate that further therapeutic strategies may be needed. And so with this, finally, I would like to acknowledge everyone that was working on this project, all our collaboration partners, and the Roma Lab. Thank you for your attention. And if anyone has any questions, I'd be happy to open the discussion. Thank you.
Hi, I'm Michael Knowles from Chapel Hill, UNC. We sometimes have trouble getting sputum specimen after we start ETI, even in adult patients that have real bronchiectasis. The way you presented the data, it implies that you were able to collect serial sputum samples after ETI in all the subjects. Is that true? If you did have trouble getting sputum, what did you do to get it?
So these patients all spontaneously produced sputum, and but therefore sample sizes reduced under ETI. We had more patients at baseline, and some dropped off because we only wanted to use spontaneous sputum.
So the data, as you showed it, comparing baseline to post-baseline, or at three different time periods, were all the same number before and after?
No, but the numbers are reduced, the patient numbers are reduced at the later time points.
I don't understand how to film the data.
Yeah, that's okay. We see your point. Thank you for it's a very nice data set, and I enjoyed reading it. So, my question was: were you surprised that there was no effect after 1 month? Because most of the effects of hydration seem to be rather quick, and the mucus clearance also happens very quickly. So,
Sit down now. Thank you for asking me.
can you please answer Scott's question?
Yes, so to answer both your questions, yes, we were surprised that we didn't see an effect because, as you've said, hydration happens, or improvement in hydration happens very quickly.
Okay.
Back.
I'll just make it. Yeah, I had the same question. Thank you for the answer. I just wondered if, one speculation might be that our 1 month samples are a bit biased because some of the patients are no longer able to expectorate, and the ones that are perhaps are biased samples. So we're perhaps skewing the data that way.
Yeah, that might be. Yeah, but as I've alluded to in my introduction, that we have these two different problems: cross-linking and dehydration. That might be that hydration improves very quickly, but somehow this cross-linking doesn't, and that this problem might persist longer. And
So building off that, your cross-linking data could be tied to your neutrophil elastase signal if you're speculating that oxidative stress is what's cross-linking. So do you think that would be part of the reason that you see a bigger change in G prime than G double prime?
yes,
Because that's cross-linking is going to dominate G double prime, and concentration, while it will affect both, will more directly affect G double prime.
we saw reduction in both, but yes, there's a bigger effect in drop in G prime. Yes.
I have one question. Your IL levels and your beta levels came back to baseline after 3 months. Do you have an explanation for why that happened?
They're still increased compared to. Wait, let me go back. So there's a large variability. So they're all reduced compared to baseline, and there's still an increase compared to healthy levels.
Okay.
And these changes here, I think just due to large variability.
Okay, and your samples dropped even for this?
Yes.
Any other questions? Thank you.
Thank you.
We've Catherine Armbruster next, who will talk to us about Pseudomonas and all things with ETI.
Hi, everyone. Excited to be here. Thanks to the organizers for the opportunity to present. Now I have a laser pointer. Excellent. Okay, thank you. I'm Catherine Armbruster. I'm a postdoc with Jen Baumberger at Dartmouth, but soon I will be an assistant professor at Carnegie Mellon, so excited to start my lab. We'll be studying CF, so stay tuned. I have no disclosures. Okay, so in the Baumberger lab, we have this longstanding interest in the upper respiratory tract of people with CF. So I thought I'd give you a little bit of an introduction to that because surely we've mostly heard about the lower respiratory tract in this meeting. So the vast majority of people with CF have sinus disease. And the sinuses are actually just another site in the respiratory tract where people with CF are chronically infected by many of the same bugs that we know cause lung infection, including Pseudomonas aeruginosa that I'll talk about later today. So we and others have shown that there's Pseudomonas and other CF-relevant pathogens in the sinuses of adults with CF. And we've also looked at, we know that Pseudomonas is notorious for its ability to evolve to persist in the lungs of people with CF. We've looked at evolution of Pseudomonas in the sinuses of adults with CF as well, and we see that Pseudomonas takes a similar kind of approach to evolving in the upper respiratory tract as it does to the lower respiratory tract. So raising the possibility that the sinuses could be not only a reservoir of CF-relevant bacteria, including Pseudomonas, but potentially even a training ground for bacteria like Pseudomonas to be inhaled from the environment and maybe first start to evolve to become more fit in the respiratory tract, even before they reach the lung. So we're interested in, when we think about the microbial populations in the respiratory tract of people with CF in our lab, we think about the whole respiratory tract. We don't just think about the lungs. And I'll talk to you about both of these sites today. And so there's a new clinical study that we have. So previously we had just looked at the sinuses, but today I'll talk to you about a study we have looking at samples from throughout the respiratory tract. And so the main questions today, we want to know how ETI impacts Pseudomonas aeruginosa prevalence and abundance throughout the respiratory tract. And then also, how does ETI treatment impact the evolution of Pseudomonas that persists post-ETI? Okay, so we have this longitudinal clinical study that was ongoing starting in 2017 that kind of accidentally became a pre-post ETI study. It was a collaboration with Vaughn Cooper, who's an evolutionary biologist, and Stella Lee, who's an ENT surgeon, and she was the person that collected all the sinus samples for us. So it's a cohort of 38 adults in the full cohort. Most of them were about 30 years old, so these are people that have probably been infected with Pseudomonas for some time. And they had at least one copy of Delta F508. So almost everybody in our study went on ETI, and we were able to compare our samples pre and post ETI. And so we collected a variety of different samples that I've kind of just summarized here. But one of the key points in our study is that the sinus, throat, and sputum samples were collected on the same day. So this is actually a huge commitment from the study participants to go to both a CF sinus clinic and a lung clinic on the same day, a pulmonary clinic. And so we used these samples to profile the microbiota. My colleague Yasmin Hilliam spoke about that yesterday, as well as look at Pseudomonas evolution that I'll tell you about today, and also to look at the kind of changes to the mucosal environment. But the samples I'll tell you about today is just, I'll show you a little bit of our 16S microbiota data just as it pertains to Pseudomonas. And then we're going to dig into the Pseudomonas evolution data set. And so to look at evolution of Pseudomonas pre and post ETI, we had taken the samples that like sinus swabs or throat swabs or sputum And we plated it on a Pseudomonas isolation agar to get all the colonies that we could grow. And we scraped up every single colony and we sequenced that entire population. So when I'm telling you today about evolution of Pseudomonas, we're not just talking about single isolates, we're talking about entire populations. So this is a better way to capture the diversity of the Pseudomonas populations that I think the field is really starting to appreciate now. And so I'll tell you about metagenome assembled genomes, which are kind of consensus genomes that we make from this population data set. Okay, so for this first question, how does ETI impact Pseudomonas prevalence and abundance throughout the respiratory tract, so not just in the lower? And we were inspired by this study from Katie Heiser when she was still in Pradeep Singh's lab, where they were tracking the Pseudomonas populations in sputum after people went on ivacaftor, so the I in ETI. And what they saw was that after people started started ivacaftor, there was this initial drop in the Pseudomonas population size, but then over time, the Pseudomonas rebounded. And so as an evolutionary biologist looking at this plot, we wondered, this looks kind of like a population bottleneck. And then perhaps the rebound is because, Pseudomonas may have evolved some trait to allow it to become more fit in this kind of ivacaftor corrected environment. And so to orient you for our study, we have samples from pre-ETI in our case, and then through about a year and a half post-ETI. So we thought maybe we could detect if there's going to be a bottleneck with ETI2, maybe we could detect that. Okay, so what happened to the microbial population? So first we looked at all bacteria by a qPCR for a 16S gene that's in all bacteria. And I'm showing you here this samples from sinus, throat, and sputum. And what we see is there's a significant drop in the population size of all bacteria. This is consistent with a much larger study, the PROMISE study that was recently published, where they also saw kind of a shrinking in the total bacterial biomass post-ETI in the lower respiratory tract. So here we can extend it to the upper respiratory tract as well. And then we also then wanted to know specifically for Pseudomonas what happened. And so by 16S amplicon microbiome sequencing, we can see that the relative abundance of Pseudomonas dramatically drops post-ETI. But if you zoom in on the post-ETI samples there, as I've done to the right of the plot, you can see that it doesn't drop to zero. In fact, only one of our 19 people where we had pre-post 16S samples, only one person were we not able to detect Pseudomonas reads by 16S. So Pseudomonas is still there. And this is also consistent with several studies at this point looking at pre-post ETI by 16S amplicon sequencing. But again, we're able to extend this observation now to the sinuses. Okay, so we wondered then, since Pseudomonas seems to be persisting, is it the same strain of Pseudomonas that's persisting, or potentially the ETI changes the airway environment so much that Pseudomonas that this person may have had for quite some time is outcompeted by a new strain of Pseudomonas that's inhaled from the environment? And so to answer this question, this is where the metagenome assembled genomes come in. And so now the sample size here has dropped because we weren't able to get mags for everybody. Basically, we had to be able to culture the Pseudomonas post-ETI to have samples here. And we have a much bigger phylogenetic tree than this, but I've just sort of summarized here some select samples from different time points from throughout the respiratory tract of the folks where we had these samples available. And so here, for example, in this phylogenetic tree that's a core genome phylogeny, so it's built on the distance between Pseudomonas strains based on 5,000 genes they share. You can see in this participant here, they have the same strain of Pseudomonas throughout their upper and lower respiratory tract over time. And so the distance between all these samples is on the order of like 100 to 200 SNPs. So it's the same strain. Okay, and so then what happens post-ETI? So the filled in purple square are samples that were collected post-ETI. And what I've done here is highlight the times when a person's pre and post-ETI samples are in the same clade. So telling us that it's the same strain of Pseudomonas pre and post-ETI. So in 6 of the 7 people, we see the same strain of Pseudomonas with quite good certainty because this is a metagenome. But in 2 people, we saw a new strain of Pseudomonas post-ETI, and so I've highlighted those here. And And so then to summarize here, so how does ETI impact Pseudomonas prevalence and abundance? So we see that there's a population bottleneck that occurs, but the same strain of Pseudomonas tends to be present in most people. So not only does ETI perhaps not totally eradicate Pseudomonas, but also in those folks where we saw maybe a new strain coming in, it might also not prevent new infections as well. But of course, this is a small study, so more work to be done there. Okay, but for those persistent Pseudomonas populations, how does ETI impact the Pseudomonas population diversity and evolution? So we know that Pseudomonas' ability to evolve in the respiratory tract, we think, is very important for its ability to persist. So we care about the population diversity and evolution post-ETI, this kind of new niche that the world has never seen before, right? Okay, so the way that we looked at this, we asked we looked at all of the mutations that were present in our post-ETI samples of Pseudomonas, and we wanted to know, of those post-ETI mutations, how many of those mutations were present pre-ETI versus how many of those mutations are newly evolved post-ETI. And so this will tell us whether, how much the airway environment has changed. If most of the mutations were present pre-ETI, then that suggests to us that Pseudomonas kind of is experiencing the same selective pressures and not differently evolving post-ETI. Okay, so I've broken down here on the y-axis, it's all post-ETI mutations, and they're either newly evolved post-ETI or they're mutations that were also present pre-ETI. And we were surprised and interested to see that across all of the folks that we had this data for, most of the post-ETI mutations were in fact newly evolved. So suggesting to us that Pseudomonas is evolving in response to a new environment post-ETI, this new CFTR-corrected environment. Okay, and so then now we wonder, can we use the mutations that Pseudomonas evolves to kind of give us an idea from the bacterium's perspective of what has changed in the airways? And so to do this, what we first did, so now you're only looking at non-synonymous mutations with encoding regions of genes. What we did was bin our mutations into the kind of functional categories based on the gene that they fell into to give us a fingerprint of what might be kind of from Pseudomonas' perspective changing in the airways post-ETI. And so here, the colors are the different functional categories, COG categories. And so, for example, these are this new inorganic ion transport and metabolism category here in this person pops up only in the new mutations. So there's selection on genes in this category that's usually genes involved in siderophore biosynthesis, so mechanisms by which Pseudomonas acquires iron from the environment are newly present post-ETI. And so I'll fill in everything. I don't expect you to look at all of these, but the idea is to just appreciate that there's kind of, the colors are different in the mutations that were present pre-ETI versus the new mutations. So suggesting that there's this different environment that Pseudomonas is experiencing post-ETI. And we can do statistics to figure out in 4 of the 6 people here, these were statistically different environments based on the functional categories of mutations. And then we could also figure out which categories were different. And so those categories are DNA repair and phage genes, motility, amino acid transport and metabolism, inorganic ion biosynthesis, and secondary metabolites. So now giving us a clue of what might actually be different in the post-ETI environment. And to dig even deeper, we wanted to know whether, okay, now we know there's maybe some shared functional categories of mutations, but what about the actual genes themselves? Is the same gene being mutated repeatedly in Pseudomonas populations infecting different people post-ETI? And this, in the evolutionary biology world, this is called parallel evolution. And it's considered really good evidence that the mutations you're seeing are actually a result of selection rather than just random chance. So we're going to look for parallel evolution in genes that are mutated post-ETI and only post-ETI. Okay, so it's actually kind of a small table, considering we started with such a huge amount of genomic data. And so what you're looking at here are the mutations that are the genes that are mutated in 2 or more people in our study. And so at the top here, there's only three genes that are mutated in 3 people here, and we can say statistically that these are unlikely to be due to random chance, so suggesting that these mutations maybe provide some kind of a fitness benefit in the post-ETI environment. And importantly, these genes were not mutated pre-ETI in our cohort. Okay, and so what do these mutations do? Okay. And so what are the functional consequences of these mutations? Are they the same across all people? So what we looked at next was site-level parallelism. And so that is even within the genes that we know are now mutated across multiple people are the same amino acids mutated, which would suggest that there's the same functional consequence of the mutation. And so for those three genes, what were they? So two of them are involved in iron acquisition from the environment. One of them is involved in kind of making sulfur bioavailable in the cell. And here's where the mutations fall on the genes. So these are lollipop diagrams. So the little lollipops show you where the amino acid change is. And so the mutations are kind of spread throughout, I would say, through these genes. And we need to go in and kind of characterize the functional consequences still. But kind of what our current interpretation of these findings is that this kind of parallel evolution of genes involved in nutrient acquisition suggests that there's probably a change in the competition for these resources in the post-ETI environment, perhaps because we know the Pseudomonas population size has shrunk so much that maybe there's a difference in selection for the ability to acquire iron from the environment. Okay, so to summarize, I showed you today that Pseudomonas aeruginosa persists post-ETI. There is a population bottleneck, but for the most part, the same strains persist. And we were able to, sometimes when we weren't able to collect sputum, we were able to continue to track the Pseudomonas population post-ETI because we were also collecting sinus and throat samples all along the way. That was kind of a fortunate aspect of our study. And then we also saw some evidence of new strains of Pseudomonas post-ETI. We don't know where they came from, but I think this is maybe an important cautionary tale, potentially. We think of Pseudomonas as an environmental bacterium, but we also know, so you can find it in water and soil, but we also know it's present ubiquitously in the built environment. And this is something I'm interested in investigating in the future, is what is the source of these new infections? I showed you that ETI imposed a population bottleneck in new selective pressures. We think that we want to kind of continue to phenotype and figure out what the functional consequences of those mutations are. But our motivation there is this idea that we can let Pseudomonas tell us through its mutations what's changing about the airway environment. I think that's a pretty exciting data set to have, especially in comparison with metabolomics and some of these other cytokine panels that we can do as well from our data set. And then in work I don't have time, of course, to tell you about today, we are also digging in more deeply into this data set of the Pseudomonas populations in the sinus, throat, and sputum, and we're looking at triggers of movement of bacteria between the two sites. And our concern is that we spend a lot of time thinking about how to eradicate Pseudomonas from the lungs and CF, but if there's still a reservoir of Pseudomonas in the sinuses, then there's a possibility that the sinuses are constantly seeding the lungs. And so we're digging into this to understand what those kind of triggers of mixing between the two sites are, including respiratory viral infection. So with that, I would like to thank my lab and collaborators, especially the folks bolded here who worked on this study directly, my funding from, especially from the NIH and the CF Foundation, and just put in a little plug for my future lab at Carnegie Mellon. So if you're interested in built environment reservoirs of opportunistic pathogens that can infect people with CF, I'd love to talk to you. So thank you.
Marianne Mullebeck, thank you for a clear presentation of a very interesting study. I wondered, the iron acquisition and sulfur bioavailability, could that also be an indication that the bacteria now are starting to grow again? And that leads to the other thought is, have you also looked at the other thing around? Are there markers that have disappeared in the remaining, such as mucoidity and signs of persistence, and now they're waking up?
That's a really great question. So one of the types of samples we saved was 96 whole plates of single isolates for phenotyping, but we haven't even dug into that yet. But I think that's a great question. We know a lot about how Pseudomonas evolves to persist in the airways, right? Pre-ETI, we have a pretty good understanding of what we think promotes fitness pre-ETI, but there's a possibility that is maybe completely untrue. And in this new post-ETI environment, some of those traits that promoted persistence pre-ETI might no longer be the case. So I'm very interested in seeing, what is the prevalence of mucoidy in this cohort compared to in a historical pre-ETI stats.
Very nice work. Basically my exact same question, except with a slight variation. Are you actually de-evolving that basically now
Yeah.
there isn't that much competition.
Yeah.
It looks
I think that's
like loss of
interesting.
function for
Yeah.
iron and all those.
Yeah. Well, so that paper that I shared at the beginning, our first evolution study of Pseudomonas in the sinuses, what we figured out was there are these in some people with CF, there's small isolated populations of Pseudomonas that evolve under kind of conditions that are very different than if you were to evolve in a large dense population where selection is the primary force driving evolution. And so instead we see a huge amount of genetic drift in those small sparse populations. And so I wonder, that was one of the things that we thought about. And after this bottleneck, are we going to maybe even see a lag time of increased drift, which can actually improve diversity in a population? And then, yeah, if the population rebounds from there, that starting diversity is extremely important in impacting future evolutionary trajectories. Cool.
Thanks.
Hey, nice talk. I'm curious to know about the evolved Pseudomonas after ETI. Are they causing trouble? Are they pathogenic, or they are behaving like commensal bacteria?
Oh man, such a great question. We haven't done it. So in Jen's lab, one of the ways we like to test for cytotoxicity and interactions with the airways is we have a polarized CF bronchial epithelial cell model, and it would be great to take some of our isolates that we've banked and just check compared to our pre-ETI, our earliest, sometimes 3 years before ETI samples, and see if there's any difference. But we just haven't gotten there yet.
Hi, Vamshi Raju from University of Alabama. Excuse me. So ETI are not really known as antimicrobial agents. So how much of these changes that are actually you're picking up in the bacteria are due to the direct drug effects versus host effects?
Oh,
Or
yeah.
host changes that are imposing these selection pressures?
Yeah, and that's a great question. I think the bottleneck, I think this is a great session to be in, by the way, because I think the bottleneck maybe is largely in that initial increase in mucociliary clearance. So I think it's not so much that Pseudomonas is evolving maybe to direct interactions with ETI itself, but the changes into the airway environment. And so one of those could be, I think also that, population size fundamentally impacts evolution of bacteria. And so I think in these small sparse population sizes, at least maybe the iron acquisition mutations that I showed you guys, I think maybe those could be related to the fact that Pseudomonas is no longer competing for iron in the environment.
And do you think these changes are different between lower and upper airways?
That's a good question. So the parallel evolved genes that I showed you guys, those evolved in both sites. But in our other study, there's a larger study that is not pre-post ETI, but from the same cohort where we're looking at movement between the two sites. We do see we've binned mutations into the mutations that move, and then there's others that can be restricted to one site or the other, and we're just digging into that. That's a great question.
Oh, thank you. Really enjoy it.
Thanks.
Great talk, Catherine, as always. Very sophisticated work. As being a sinus surgeon, I just have some technical questions. I call the lung as the lower sinus. And my question is I mean, it's a really great concept. I always think that there's silent aspiration every day, even if it's a normal patient. So there's always sinus microbiomes can go to the lower lungs. Michael, when did you collect the post-ETI? Because as usually it's like sputums, we don't need a lot of patients that has a lot of mucus. Is that a routine visit or patient exacerbated?
Yeah, I kind of sped through the study design, but so they were regularly scheduled visits, but then there were additional visits if there was a sinus or lung exacerbation, pulmonary exacerbation. But yeah, it was quarterly visits, and we like everyone else experienced a drop off in sputum over time.
Is anybody on antibiotic rinses and their
Yes.
sinuses?
Yeah, we have all that data. Yes, and anti-inflammatories. We have all that data and haven't exactly dug into it yet. So Yasmin Hilliam, who's doing the microbiota side of this study, she's starting to overlay that data with her data set and see if there's any relationships. But I certainly haven't dug into it yet. Okay.
That was a great study. I have one question. Do you have any correlative clinical data for exacerbations and what happened? Did they stay the same, or did you see a decrease when the genetic evolution happened?
We have that data. I haven't tried to overlay it with this data set yet. That's a good question.
Okay.
I mean, so in our cohort, when people went on ETI, they felt a lot better. So we've definitely seen that, but I haven't looked specifically for these mutations.
Okay, thanks.
Thanks, everyone.
All right, next we're going to have Daniel Arajades from Dartmouth, I believe. If my geography is correct. We did not plan that to be two back-to-back. But for those in the back, there are chairs in the front. So if you like standing, stand. If you'd like to sit, come sit. So take it away, Daniel.
Thank you. Okay. All right. Thanks, everyone, and thanks to the organizers for inviting me to present some of my work. As mentioned, my name's Dan Aragides. I'm an adult pulmonologist and a bench researcher as well at Dartmouth, and I'm going to be presenting some of my work on the effects of ETI on primary lung macrophages. I don't have any disclosures related to this presentation, so I don't think that it's going to be controversial if I say that highly effective modulator therapy has really fundamentally changed the landscape of care of people with CF who are lucky enough to be on them. And the effects of ETI are primarily due to the effects on the airway mucus, improving the hydration of mucus, as we've seen, increasing expectoration of bugs and mucociliary clearance. But as has been mentioned by, I think, pretty much all the presenters today so far, this has unfortunately not been associated with a return of sterilizing immunity to the lung, or sterilizing in the sense of, getting rid of the bad actors in the lung, including Staph aureus and Pseudomonas, as seen by the PROMISE micro study here, as well as multiple other studies. And with me and my background in immunology, the questions that raises for me is how does that impact host pathogen interactions in a post-ETI world? So alveolar macrophages or lung macrophages serve as the sort of sentinels within the lung. They're situated within the alveolar space and are the first poised to respond to infections within the lung, and therefore represent a good place to start when you're thinking about what are the consequences on host pathogen interactions. As mentioned, they serve as the first line of defense in the lung, but it's also known that CF macrophages have altered pathogen responses, and those have primarily been shown to be hyperinflammatory and with poor phagocytosis. On the other hand, CF macrophages have also been shown to respond to CFTR modulators in a beneficial way, returning them more towards a baseline status of decreased inflammation and improved phagocytosis. And to sort of highlight some of these background data, we know from John Engelhardt's work with the CF ferrets that there is impaired innate immunity in CF, even when people are on lifelong antibiotics and there's no detectable infection there, they still show pronounced inflammation and mucus. And from other works with mouse bone marrow-derived macrophages, CF primary human monocyte-derived macrophages, and a knockout of CFTR in human monocyte-derived macrophages, they've also been shown to exhibit increased inflammation and poor phagocytosis and killing by multiple different groups. But they can respond to CFTR modulators. So the first evidence of this was shown by Bruce Stanton's group and Roxana Barnaby in 2017 with the early generation modulators, lumacaftor, ivacaftor. When they were added back to CF macrophages in vitro, they improved their phagocytosis and killing. And multiple other groups have shown similar phenotypes, including with the newer generation modulators, ETI, including our report in just this year. There's also been a couple of studies on PBMCs with pre-post ETI studies, as well as add back of ETI in vitro showing improved responses. But one thing that you'll notice is missing from this landscape here are studies on primary human lung macrophages, and there's a good reason for that. They're harder to get, and obviously peripheral blood samples are much easier to come by, but it doesn't necessarily mean that they're going to respond the same way. So we undertook some studies where we actually performed a research bronchoscopy on volunteers with CF as well as non-CF volunteers. And these were people who were at baseline, not in exacerbation, with mild to moderate CF lung disease. And at the same time, we isolated peripheral blood to get peripheral blood monocytes and differentiated them into monocyte-derived macrophages, or MDM, with MCSF for 7 days in vitro. We then plated the cells, treated them with CFTR modulators for 48 hours in vitro versus controls, sort of in parallel. and then performed some downstream assays in them. And those downstream assays included phagocytosis assays via a gentamicin protection assay using both cell types, and we used Pseudomonas aeruginosa for those experiments. We also measured their metabolic responses because we know that metabolism in macrophages is very critical to their ultimate function. We measured cytokine secretion as well as some early data on transcriptomic responses in the macrophages that I'm going to show today. So as we published this summer, when we looked at phagocytosis of both a laboratory isolate and a clinical isolate of Pseudomonas aeruginosa in CF macrophages, and these are MDM studies, we saw a nice kind of dose-responsive increase in phagocytosis where the double modulator tez-iva combination, or TI as shown on the slides there, had an incremental increase in phagocytosis efficiency, and then there was an additional benefit with ETI in both of the strains studied. And this was nice. It kind of fit into the overall kind of landscape, showing that, you know, the CF macrophages do better when you give them back the ETI. One of the things that was a little bit surprising when we looked at it, however, was when we took non-CF MDMs, we actually saw a similar response, and there was actually no statistically significant difference between the two cohorts in their response to ETI. So that was kind of cool and interesting. You know, you'll notice there is some variation between the groups, but overall they still responded favorably to ETI. And I should say for these studies, there is inter-experiment variability, but the lines connecting dots represent a single experiment from a single subject. So you can see that the within subject variability is actually, there's fairly low, and it was pretty consistent on a single assay. When we then looked, however, at primary lung macrophages, we found something very different. And this was combined from multiple different Pseudomonas strains, which is why you'll see the higher N there. But I combined them all because there really was no difference between the strains that we saw there. And whereas there was maybe a small increase in phagocytosis with the double modulators only seen in the upper lobe macrophages, there was actually a strong inhibition of phagocytosis when we added the elexacaftor back in. And this was really surprising to us. But it was, as you can see, very reproducible with multiple different subjects, multiple different experiments, and multiple strains of Pseudomonas. We saw similar effects with the non-CF lung macrophages, and those all showed a kind of a decremental effect of the ETI on phagocytosis with the primary lung macrophages. So that certainly raised some eyebrows and some questions, and so we wanted to dig a little deeper and try to find out what is the underlying the different response to modulators between the two cell types. As we know from a decade of or more of work, metabolic effects, metabolism in macrophages is sort of intimately intertwined with their function. And you can see on the left, inflammatory macrophages, or sometimes called M1 macrophages, tend to rely more on glycolysis for ATP generation, even in the presence of adequate oxygen, whereas anti-inflammatory, or sometimes called M2 macrophages, tend to rely more on oxidative phosphorylation. And you can actually shift the sort of function of a macrophage by perturbing their metabolism. So this is sort of a two-way street. And we measure this by something called a seahorse assay, which measures oxygen consumption as a proxy for mitochondrial respiration. And then it can measure glycolysis at the same time by measuring acidification. And this is an example of what one of these plots looks like from a single individual. The drugs are added sequentially down the line there to measure different parameters. I'm not going to have time to go into all of that today. But what you can see is that at the baseline, the triple modulators had a strong inhibitory effect on mitochondrial respiration in the CF MDMs. And this was seen, so that was just a single individual. When you pool data from multiple individuals on the right here, you can see that that was very consistent between multiple different subjects. And again, no significant difference seen between CF and non-CF donors. So what about the lung macrophages? Could there be different responses in their metabolism that underlie the different phenotypes that we saw with the phagocytosis? And the answer was no. They actually responded again very similarly to the triple modulators, whereby there was a strong inhibition of mitochondrial respiration in the lung macrophages with the triple modulators, but no effect of the double modulators, both in the CF and non-CF cohorts. Next, we looked at inflammatory cytokine production. So this was a infection model on cells that were either pretreated with DMSO, Teziva, or ETI. And you can see that the CF MDMs here strongly upregulated multiple inflammatory cytokines. Not surprisingly, things like IL-1 beta, IL-8, TNF-alpha, MIP-1 alpha, and MIP-1 beta were all strongly upregulated in the presence of Pseudomonas. However, there really was no significant effect of the modulators on inflammatory cytokine production, at least in this setting. And this was, a little bit counter to some of the prior studies, although the more recent study by Ben Kopp's group that used the triple modulators also found a similar result, which was somewhat reassuring. The non-CF MDMs responded similarly, and really very little difference, as you can see here, between the two cohorts. When we looked at the lung macrophages, however, they had a much more restricted sort of repertoire of inflammatory cytokines that they produced. You could see that IL-8, TNF-alpha, and the MIP proteins were again prominent in what they were able to produce. But the lung macrophages actually did downregulate IL-8 and TNF-alpha significantly in response to the ETI treatment, in contrast to the MDM. And overall, you can get a sense that maybe they're not quite as primed to be inflammatory as the MDM are. When we look in a little more detailed manner at the transcriptomes from these cells, this was an experiment where we took MDM and lung macrophages from one subject with CF and one subject without CF. Sorry, the key got kind of skewed down there. And then we pre-treated them with modulators and then infected them with two different Pseudomonas strains. And what I really want to highlight here is that when you do unsupervised clustering on the transcriptomes from these cells, they really segregate out pretty strictly by macrophage subtype on the second line there. Whereas the subject and cohort in this case, because one of the subjects had CF and the other did not, didn't segregate out as much there. And so really the strongest difference between the transcriptomes was the cell type of origin. So a lung macrophage from an uninfected non-CF subject was more similar to an infected CF subject than it was from a monocyte-derived macrophage from the same subject. So finally, for the last part of this talk, I'm going to shift a little bit. And some of these unexpected findings that we found raised some questions about, are we looking at the right experimental conditions? And one of the questions that comes up is, what is the optimal concentration of modulators to use in vitro for these experiments? If you look through the literature, the concentrations that people use actually varies quite a bit, sometimes even within the same papers. Infiltrating monocytes are likely to be exposed to serum concentrations of the modulators while they're in the peripheral blood, but as they transition from that to the alveolar space, the concentrations in the epithelial lining fluid is really unknown, at least as far as I could find from the literature. Maybe someone here has some unpublished data that they know better than I do, but I couldn't find anything personally. There were a couple of small studies on sputum concentrations of modulators, but that was about it, and only with the earlier generation modulators. And finally, we know that ivacaftor can destabilize rescued CFTR. That's been shown by multiple different groups. And in the early studies from Bruce Stanton's group, it was shown to inhibit phagocytosis at higher concentrations. And for that reason, up until this point, we've used ivacaftor at a low concentration of 30 nanomolar with tezacaftor at 3 micromolar and elexacaftor at 6 micromolar. But we decided to go back and be a little bit more rigorous about sort of finding the optimal concentrations, and then looked at 4 different concentrations of ivacaftor, 2 of tezacaftor, and 3 of elexacaftor, all sort of in parallel in the same studies. And when you compare the MDM and the LM responses to this, you can see that there is quite a bit of variability depending on which modulator combination of concentrations you use, but also that there is a marked difference in the responses between the two cell types to the different concentrations of the modulators. And one way of visualizing this, so graphed here, I've put ivacaftor on the x-axis and then shown grouped all the different combinations of the tez and the elexacaftor concentrations. And what you can see, again, that there's a pretty marked difference between the responses of the MDM and the lung macrophages to the different concentrations of ivacaftor. But you'll notice that all the lines basically more or less parallel each other, arguing that there's some independence to the dose response there. The lung macrophages in particular didn't like the very high concentrations of the ivacaftor. The tezacaftor results were a little more mixed. We only used 2 different concentrations of this due to sort of constraints on the numbers of cells that we can have. In general, it was positive, but in combination with the higher elexacaftor concentrations, it did sometimes have negative effects. And then the elexacaftor effects were more interesting, where the MDM had the best effect to the lowest concentration, and then kind of tailed off in the lung macrophages in general, did worse the more elexacaftor that you gave them with their phagocytosis. So in summary, we've shown that the monocyte-derived macrophages and lung macrophages respond differently to modulators, and at the transcriptome level are in fact very different from one another. That the modulators appear to have negative effects on lung macrophage phagocytosis at high concentrations, and that the CF and the non-CF subjects actually responded very similar to both of those. So I'd like to thank lab members, statistical support, and my mentorship committee for this, and my funding sources. And with that, I'm happy to take any questions.
Thanks for a great talk. I guess a simple question. Do you think the kind of paradoxical or surprising effects on phagocytosis are direct effects on CFTR function, or is it sort of an off-target effect?
Yeah, I mean, we've thought a lot about that question. I don't have the data to suggest one or the other yet. I think there's some circumstantial evidence that points to the fact that it may be actually off target. One being the fact that the CF and the non-CF macrophages both responded very similarly. The level of CFTR in the macrophages is very low. Some people don't believe that it's there. We don't actually detect any CFTR transcript in the RNA-seq data. So I have a suspicion that it may be off-target effects, but I don't have proof of that yet.
And I guess kind of related, my brain works very slowly. Were you able to test elexacaftor as sort of a monotherapy to differentiate, is it the triple versus the double, or is it just an elexacaftor issue?
Yeah, so we do that as part of those combinations of the drugs that we use that I kind of sped through at the end there, so sorry about that. And it behaves very similarly. So the elexacaftor monotherapy there would be the blue line, so 0 of IVA and TEZ, and it looks pretty similar to the rest of the groups. I haven't tested it alone in all of the assays that we've done. That's something that we're going to be, working on. But as you sort of noticed, the elexacaftor seemed to be kind of the wild card here that sort of behaved differently from the other modulators in a lot of the assays that we did.
Alessandra W34 from UNC. My understanding is that these drugs accumulate in the tissue. Is it known how much ETI is specifically in lung macrophages in patients?
Like inside the lung macrophages?
Inside, if it's accumulated.
I don't know the answer to that question. I don't know if anyone else does. Like I said, I've seen a couple of studies that looked at sputum concentrations, but only on people on the earlier generation modulators, and it looked like it was on the order of 10 to 100-fold less than it was in the serum in some of those patients. But, sputum is not necessarily the same as what is in the airways, obviously.
I wonder if that might help you define the right concentration to treat them in vitro.
Yeah, it would be great if we could try to figure out, what is the actual concentration within the epithelial lining fluid.
So casting this on to patient care, if you improve MAC phagocytosis, how do you anticipate that's going to help ETI patients feel even better? What's the solution to this at the end of the day?
Presumably it would help to decrease the bacterial burden within the lung by helping to clear them out and sort of changing that balance between, the amount of bacteria within the lung. It's a little bit speculative, obviously, and there's no real way to test that in vivo that I can think of. But I think that it would, potentially, there's some signal here that may potentially explain why bacteria persist in the lung if the phagocytosis is impaired, and clearing out the infection is obviously the goal that we all have.
Okay.
All right.
Thank you,
you.
Daniel. We have Dr. Engelhardt next to talk to us about CFTR mRNA and the ferret airway.
So advancing.
You can hit that, or you can just use this.
Pointers on there.
Or,
Right here.
okay. That.
So this represents that.
Cool.
How many minutes when it turns yellow? It turns yellow. 1 minute.
3.
When it turns yellow.
But you can, we have plenty of time.
Okay.
Half an hour, so-
Okay. All right. Great. All right. I'd like to thank the organizers for the opportunity to speak today. I'm going to be talking about a story of evaluating a lipid nanoparticle and its efficiency in CF ferrets. There we go. All right. These are my disclosures. The one relevant, we do have a service contract with Arcturus, which is the nanoparticle I'm going to be talking about, and I'm a consultant as well. So Arcturus has developed a lipid nanoparticle with an optimized, chemically optimized mRNA to human CFTR and codon optimized, with the goal of delivering this to CF lungs and repairing the basic defect in the lungs. Now, when considering gene therapy for cystic fibrosis, I think it's important to put this in the context of what the target cells are. And throughout my talk, I'll be referring to those cells, the functions of what is known for some of these cell types, CFTR and the expression, and where they're located. So there are a number of different trophic units within the lung that are composed of unique types of epithelial cells. Now, of these, 5 express CFTR, goblet cells, ionocytes, transitional basal cells, club cells, which are mainly in the distal airways, and alveolar type 2 cells, which are down in the gas exchanging airspace. Now, it's currently unclear what CFTR expressing cell types in the airway must be targeted by mRNA therapy or any other gene therapy in order to correct the basic defect. And we've developed a number of different cystic fibrosis ferret models at Iowa, initially a CFTR knockout. I'm going to be talking about a G551D knock-in model. Both of these develop mucus-laden airways and bacterial infections, including Pseudomonas, with progression of disease. And importantly, this G551D ferret is responsive to the CFTR potentiator, ivacaftor, I'll call VX770. And it's really unclear, this model is useful for looking at both the onset of disease and potential reversal using gene therapies or RNA therapies. So we began our studies using a transgenic ferret model that I refer to as ROSA-TG that switches from tomato expression to EGFP expression upon Cre activation. We took primary basal cells, polarized these, and then transfected them with the LUNAR encoding Cree mRNA and saw efficient conversion of airway cells at the air-liquid interface. Now, we then looked at CFTR correction using a human CFTR, either vehicle or LUNAR with the CFTR mRNA, comparing here transfection into knockout airway epithelial. One can see no CFTR following transfection, CFTR expression, delta F 508, ferret, airway epithelium, very little CFTR because it's not processed. Then we see good CFTR expression. And in wild type, we also saw a slight increase in expression following transfection with the mRNA. And importantly, the functional correction here in both CFTR knockout and delta F following IBMX4 scolin or inhibition with GlyH101 that blocks the CFTR channel, we can see a significant amount of correction. We then went in vivo to evaluate Cree mRNA loaded into LUNAR in the Rosa TG ferrets. There was efficient transfection and conversion of this digital reporter, both in the trachea as well as in the interpulmonary airways. If one quantifies this in the trachea and interlobar airways, it's approximately 30 to 40% of the cells convert. We also tested tomato mRNA loaded into LUNAR. These are in wild-type ferrets. You can see very good expression in the surface airway epithelium, as well as even this gland duct, as you can see here in the interpulmonary airway. And importantly, similar transfection approach, but using the human CFTR mRNA loaded into LUNAR, comparing vehicle or human CFTR transfected airway epithelia. One can see here 2 ionocytes, I'll be talking about these later, that express very high levels of CFTR, potentially a secretory cell expressing CFTR in the vehicle treated, but significantly more expression along the apical boundary here of the surface airway. Looks predominantly like ciliated cells as co-stained here for acetylated tubulin. So what cell types are transfected by the LUNAR lipid nanoparticle? Well, we phenotyped this at first using the tomato mRNA loaded into the LUNAR. Here, co-expression, or looking at tomato expression with FOXJ1, a ciliated cell marker that marks the nucleus, and MUC5AC marking secretory cells. And you can see predominantly ciliated cells are positive for the tomato. If we look down here on the bottom, see similar, maybe an example of 2 secretory cells here marked in yellow, where you can see very faint expression that appears to overlap with the MUC5AC staining, but predominantly most of the secretory cells appeared negative. Now, how about pulmonary ionocytes? So pulmonary ionocytes exist within the proximal conducting airways, the cartilaginous airways of the lung. They're associated with submucosal glands in these particular regions. And interestingly, pulmonary ionocytes express about 50% of CFTR, but they're only 1% of the epithelium in this region compared to the secretory cells that are approximately 30% express CFTR. So we've shown recently that pulmonary ionocytes that express very high levels of CFTR at their apical surface are important for pH regulation of the airway surface fluid, ASL, airway surface liquid volume, its viscosity, and in the absence of pulmonary ionocytes here in a FOXI1 knockout ferret, one has reduced clearance and all these other parameters that I've marked here. So our pulmonary ionocytes transfected with the LUNAR LMP. So we first did the experiment looking at human CFTR expression. Obviously, this is going to be difficult here to distinguish between endogenous CFTR and ionocytes, where you can see right here 2 ionocytes in a vehicle transfected, marked with ATPase here in green. And again, we saw CFTR expression within the LUNAR human CFTR transfected, but in addition to other cells labeled apically that were not ionocytes. So if we do this in a more directed way, evaluating tomato expression, one can see two ionocytes here that are not transfected. Perhaps an ionocyte here that looks like it has a little bit of the ATPase expression and tomato expression, but not extremely impressive example. So we took the digital reporter approach using the ROSA TG animals using Lunar Cree transfection. Here we've marked MUC5AC in red, BSND in magenta. And you can see here an ionocyte that potentially is positive. We look over here, it's kind of blown up for the one right there. And it's questionably though, because this is a membrane-bound GFP and we don't see this membrane expressing. Here's a very clear negative ionocyte that did not convert to EGFP. A couple of other examples we could see that did look more convincing. For example, here's an ionocyte broken out into two channels. Clearly looks like the basolateral membrane's labeled, and here's another example of one that's positive and one that's negative. If we look at other cell types, again here, P63, which marks basal cells. Not surprisingly, basal cells don't access the apical boundary. There are very few basal cells that converted to GFP. And secretory cells, as I had mentioned earlier, are predominantly negative. But when you do see a positive secretory cell, it's pretty obvious. You can see GFP in the cytoplasm and enriched on the basolateral membrane, and here's a negative secretory cell. So Lunar LMP complexes efficiently transfect ciliated cells and some ionocytes, but very few secretory cells and no basal cells. And so ciliated cells don't normally express CFTR, and it's highly controversial whether or not if you complement or overexpress CFTR within the ciliated cell, can you complement disease? And so we want to ask whether or not the Lunar human CFTR mRNA could complement the mucociliary clearance defect in cystic fibrosis ferrets. So we turned to an assay which is fairly robust that looks at mucociliary clearance using PET-CT and a gadolinium-68 conjugated to albumin. Here, the radioactivity is deposited, sort of sprayed in right at the carina, enters into the large bronchi and the distal end of the trachea, and then we quantify clearance. And you can see here in a wild-type animal, this is going from distal to proximal, very rapid clearance up to the proximal end of the trachea. This is a CF animal maintained on ivacaftor, VX770. Clears very rapidly, and here's one off that is clearing much more slowly. And you can quantify this. This is just an example of a set of ferrets that were measured at baseline on VX770 here, maintaining about 80% maximal clearance, then taken off for 1, 2, 3, or 4 weeks, and then put back on VX770 for 2 weeks. And you can see gradual decrease in mucociliary clearance and then bounces back up when it goes back on VX770. And you can quantify this using the rate of clearance within the first, typically we're using about the first 6 minutes, going baseline, going off VX770, back on. And so airway clearance, at least in this model, can be indexed and is CFTR dependent. And so we turned to do this study with Lunar, where we took G551D ferrets, removed them from VX770 till they had a moderate MCC mucociliary clearance defect of about 20-40%. We treated with Lunar, human CFTR mRNA or tomato as the negative control, and then assessed clearance 24 hours later. And the results were quite impressive. You can see here in the group that was treated with Lunar human CFTR. These are the ferrets prior to treatment with Lunar CFTR. This is following treatment in the same 8 animals. And these are the 6 animals treated before and after with the tomato. And if one quantifies the first 6 minutes, we can use area under the curve or rate. We're tending to use rate now. One sees here's baseline in the 8 animals treated with the human CFTR after human CFTR Lunar treatment, increase in the rate, no increase in with the Lunar tomato. And these differences are statistically significant, and the baselines are very similar. This is just projecting the data on a per animal basis. Every animal had some extent of increase within the lunar human CFTR group and variable changes within the lunar tomato group. And now, I see this through a lens that this is particularly interesting and important to me because we know that if we knock out ionocytes within a ferret, one sees decreased mucociliary clearance. So these are FOXO1 knockout here compared to wild type in CF animals either on VX770 or off VX770. And so the fact that the lunar transfects predominantly ciliated cells, yet we're seeing this enhanced clearance is very interesting. So if one thinks about what this actually means and what we've actually potentially demonstrated, there are some caveats to conclusions that ciliated cells can complement. And these are as follows. So in a wild type animal, we know if we're using tomato expression as an index for the level of transfection, certainly ciliated cells are more highly transfected. But secretory cells have very low level expression, potentially below the threshold of detecting the tomato fluorescence. And secretory cells normally do not express significant amounts of CFTR. So it's unclear whether or not we're indexing all the cells that are transfected. Second, one would assume if you go to a digital reporter using Cree, that switch might be much more sensitive. And indeed, we can see very convincing secretory cells that have been transfected with Cree, but again, predominantly the ciliated cells are the cells that are converting the reporter. And so it's unclear whether or not low-level Cree transfection is also limiting the sensitivity by which to detect the percentage of secretory cells that are transfected and whether or not they're contributing to the complementation when using the human CFTR. If one makes the analogy to a Cree driver, certainly the level of expression of the driver gene in which Cree ER2 is knocked into impacts the efficiency at which it can label that cell compartment. So to summarize, lunar LNPs have effectively delivered human CFTR and corrected both the electrophysiologic defect as well as the clearance defect in CF ferrets. The improved MCC, we saw improved MCC, but the highest level of transfection were ciliated cells and lower in the secretory cells and ionocytes, but it's unclear the contribution of each of these cell types has to the correction of the mucociliary clearance defect. Some of the answers to this are really going to involve a requirement to have genetically engineered ferrets that we can do those types of reconstitution experiments in an animal genetically using Cree. And CF and other transgenic ferrets, I think we've shown that they have high utility in a number of different ways to try to understand cell types that are targeted as well as the mechanisms of complementing disease. And Arcturus currently is moving the lunar CFTR into phase 1 trials to evaluate. I'd like to thank, projects like this using larger animal models are really, it takes a town. I'm giving the presentation for Dr. Xiaoming Liu, who couldn't attend, and I'd like to thank those at Arcturus that contributed, the funding agencies. Thanks.
If I may have a question, thank you for the great talk and a very interesting, model with the ferrets. Just one quick one. So I'm just curious the kinetics after this transfection for the ferret with this defection. After you knock in or transfect with the LUNA or CFTR, we can see the CFTR function. I'm just curious what's the kinetics after that, like day 2 or day 3. Is the CFTR function still there, or what's the half-life?
Yeah, excellent question. So we haven't done the durability experiments yet. They need to be done, where we transfect and then assess for functional correction at various days afterwards. We know from at least the Cree transfection that the conversion of the reporter is extremely rapid. So within, 24 to 36 hours, you've reached the maximum amount of conversion. And that's one of the reasons we chose a short time point to initially evaluate the human CFTR.
Got it. Thank you.
John, as always, great work. Again, emphasizing the beauty of these models. So I'm curious, clearly you see cell type differences. Originally, I was expecting this to be like where, depending on the native proportion of cells, you would see a transfection, but clearly that's not the answer, even for LMPs, which are not known to have cell type specificity. So that raised the question in terms of, what do you think makes this so differential compared to the types of cells? Is it the cell properties of taking up these LMPs, or is it the location of where those cells are present?
You know, I wouldn't expect an LMP to be taken up efficiently by a ciliated cell with everything moving on its membrane. I mean, I don't know, you know, why, and I don't think it's specific just to this LMP. I mean, I think others are seeing similar things with other LMP formulations. I'm not a chemist, an LMP chemist, so I can't say why, you know, they're attracted to ciliated cells. But
you're the best cell biologist I know, so I was hoping you'd talk about the cells. So thank you.
Yeah.
Very interesting presentation, John. Thank you so much. Can you comment on a pre-treatment maybe that you have done on the ferrets before you did your transfection? Do you have to remove the mucus, or the LNPs are able to penetrate that mucus layer? And were you able to show that?
Yeah, this is a really good question. So these studies in the ferrets, I mean, they were to get a 20 to 40% mucociliary clearance defect. You're talking about pretty early phases of pathophysiology, like, you know, 2 months off VX770, not, you know, a year with permanently colonized and a mucus-laden airway. You know, I know from testing other vector systems, I mean, when you get a mucus cast in the airway, nothing is going to make it through. And, you know, we didn't precondition these animals. We didn't use a mucolytic. But it's very clear to me that there's going to have to be that approach for very progressive disease that's significant and where animals are permanently colonized. So we don't know the colonization status of this. I'd say they're very early if they, you know, do have bacteria by the time we went in to do the MCC.
Just curiosity, can you do bronchoalveolar lavages in ferrets just to flush things out? Okay.
Yeah, we do a lot of that.
Thank you.
Yeah.
Congratulations on a great talk and also the recent mechanistic ionocyte data. I have a question related to the MCC, and maybe I missed it. Have you also measured MCC in wild type animals? And how does it compare then to the mRNA treated?
Yeah, we did measure wild type animals. It's on this. It's this blue little thing right here. So, I mean, I don't have them all plotted on the same graph with the lunar human CFTR, but the lunar human CFTR is actually a little higher than, you know, even CF on VX770. So it's comparable to wild type, like full correction.
So, because Scott is looking at me, maybe a question to him. So how does that compare to your ETI studies in patients?
Thanks. I needed a question. It's a complicated answer. You know, John's studying the trachea, we're studying the whole lung. What we actually see, which is kind of, you know, disturbing maybe, is that if you look at an untreated CF patient compared to a healthy control, things like central lung clearance are not abnormal. You know, we really see a mucociliary clearance defect in CF patients distal in the lung in the periphery. When we put a CF patient on a highly effective modulator, whether it's ivacaftor or ETI, we actually superficially it appears like supernormal clearance. So it's not just reaching to normal levels, but it's supernormal. We think that's a function of the airways disease. In a CF patient, when they inhale isotope, we put more tracer in the central airways where clearance exists. Whereas a healthy subject, that tracer is going much deeper in the lung where clearance is intrinsically slower. So it's hard to make sort of a healthy to CF comparison.
Yeah, I will make one comment that, you know, these ferrets with on and off ivacaftor, I mean, they're homozygous G551D. So if you look electrophysiologically, which I didn't show the data, in a air-liquid interface, you get 75% of wild type when you treat with ivacaftor. So this is the best case scenario with the CFTR modulator treatment in an animal that's homozygous for G551D. Yeah, David.
I'm David Geller from Arcturus. So thank you, John, for presenting this work. It's wonderful data. As a simple clinician, I usually go for the bottom line, and I looked at it when I first looked at this data, I thought, wow, that really works. It was a very dramatic increase in mucociliary clearance in the trachea, coupled with the data that Javier Campos Gomez presented on Thursday with the HBE data showing wild type activity after lunar CFTR with a chloride current, and Trikafta-like activity without a chloride current in vitro. I thought that was, coupled with those in vitro and in vivo findings, it was very compelling evidence to take into the clinic, which we have done and completed the healthy volunteer portion of the phase 1 study. So I'm happy to announce that the Safety Review Committee has said, yep, you're okay to go into patients. We've already started screening, and so we're looking forward to seeing what happens there. But to me, as a clinician, bottom line is, hey, this stuff works. And the scientific discussion about why it works is fascinating. I'm just happy it works. And so that's the bottom line, 2 words, it works. So it's not an advertisement. But I did have some questions. I think, in vitro, when you do the immunohistochemistry, it looks like lunar CFTR transfects all the cells, almost evenly, like a third of the cells to 40% of the ciliated cells. And I'm not sure what the difference is between the in vitro wells and in vivo. So comment on that. And then the second question is the recent data from University of Iowa showing the ionocytes actually can work in reverse and suck water back into the cells as opposed to pumping it out. Trying to figure that one out in my head too. I didn't know if you had an answer for that.
Well, I can take another 30 minutes and go over that, but I think that the two studies have differences. I mean, we show that they both absorb fluid. The McCrae, study showed that they were not important in secreting, and we showed that without ionocytes, you get a dehydrated airway epithelium with reduced pH. So I think there are some explanations for why there are differences. Culture systems being a big one. The fact that there are multiple types of ionocytes, probably functions of ionocytes are partitioned between different subtypes. But I think ultimately that's going to inform even more, of how ionocytes function once we figure out why there were differences.
Great. So since you're showing that it works, the question is how long will it work for? How often do you anticipate you'll need to dose the animals?
Yeah, we're entering into some studies, shifting, more to electrophysiologic complementation of the trachea, but using oozing chambers to look at the durability in vivo. There have been some studies in vitro. I haven't done those studies, but and maybe David could comment on them. I've heard about them from the CFFT labs that. And this may just be LMPs in general, but half-life on the order of 3 to 5 days for CFTR function, like electrophysiologic function, which is a lot longer than I thought it would be, and that's super encouraging. Yeah.
You don't need me. That was the right answer.
Okay. Yeah. Great. Thank you.
Thank you.